Thank you everyone for coming, and I see unfamiliar faces, which means that's the integrated applied mathematics people. I'm very pleased for that because groups tend to be quite tribal. CCOM only does CCOM stuff, and IM only does IM stuff. In fact, the nature of the integrated applied mathematics program sort of fosters that sort of collaboration. And Gabby is the first student that we've had here at CCOM in the IM program, which is really quite a useful thing for us because this type of skill set that she brings by having more of a pure mathematics background adds a nice set of skills and knowledge to what's going on over here. So quite pleased to see you all. The nature of this, in fact, proposal defense, we don't really do that in ocean engineering, which is where most of the CCOM students are. So this is really satisfying the requirement of the IM program for a public defense of the proposal. The format that we'll use is Gabby will talk for about 45 minutes. She has given each of her committee members a research proposal, which we all have. And the nature of this is that Gabby will talk for 45 minutes. We'll then open up the questions to the public because afterwards the committee members will be meeting with Gabby alone. So committee members, hold your questions till after everyone else leaves. Everyone else holds your questions until Gabby finishes, and there will be plenty of time to talk about that. So with that, I will turn it over to Gabby.
Yay! All right, hi, hello. Thank you all for being here. Like Kim said, it's really nice to kind of see a mixture of my worlds, of my math and my CCOM friends. So thank you for taking the time out of your day to be here. And I am really excited to talk about some of the research that I have been working on and am going to continue to be working on, which is exploring coral reef monitoring using machine learning and ICESat-2. So, Christopher.
Take the mouse and click in the window where your presentation.
Awesome. I'm so glad you're here. So to get started, can anyone tell me what this is a photo of or where this photo is taken? Any ideas? I know there's at least 6 of you here that know what this is. Somebody, anybody?
Honduras?
Honduras, that's a good guess. Very similar idea.
Guatemala?
No. Close.
In Australia.
It is in Australia. Thank you. So this is the Great Barrier Reef in Australia. It is one of the 7 natural wonders of the world, and it is actually the largest coral reef system in the world. It is home to a diverse abundance of life. There's like 400 different types of corals, 1,500 different types of fish, sea turtles, dolphins, whales, and so much more. So I'm really sad to tell you the coral reefs are actually dying. And unfortunately, this is largely due to a number of climate change related reasons. For one, the ocean temperatures are rising, the sea levels are rising, and we're seeing more severe and more frequent weather storms. And this is leading to things like mass bleaching events, which we can see in this image on the left. Spread of infectious disease, a lack of food for the corals, and an overall destruction of the reef structure. So because of this, it is imperative that we step in and take measures to monitor these coral reefs in order to prevent complete devastation now. The current techniques that we have for monitoring coral reefs are really expensive and require you to be in the ocean and on site. For example, here's a couple ways that we are currently monitoring coral reefs. For one, this top middle photo is what we call a manta tow survey. So a diver holds onto a board, is towed by a boat, and the board takes data while it goes over the corals. Another example, which we can see with our friend Dr. Jen Dykstra here in the top right-hand corner, is what we would call a fixed site survey. And in this case, Dr. Dykstra is taking photos of the coral reefs. Another example of a fixed site survey is of visual counts, where we can see our friend Glenna Dyson, who is doing things like measuring corals and probably counting the amount of corals there are of each species, of each size, stuff like that. So like I said, all of these different methods require us to be in the water. So is remote sensing the solution? Remote sensing is this idea that we can collect data without actually needing to be on site. Perfect, right? So it's collecting data from satellites, from boats, from planes. But remote sensing is not new to the world of coral reef monitoring. In fact, it first shows up in the literature in 1975, and this author is utilizing a Landsat image to do a geomorphic zonation of a coral reef. Since then, the limitations and advantages of coral reef monitoring and research in general using remote sensing has been widely researched. But most of the literature is utilizing multispectral and hyperspectral imagery, which is actual images of the Earth from a satellite. So let's talk about ICESat-2. ICESat-2 is a different type of remote sensing data. It was launched in 2018 for the purpose of collecting data about the cryosphere. So essentially, can we monitor changes in the ice caps in the ice sheets? But ICESat utilizes a green photon counting LiDAR, and this is really important for two particular reasons. For one, it is a LiDAR. LiDAR stands for light detection and ranging, which utilizes pulses of light beams, of lasers, to capture elevation or depth data from them. The other really important aspect of this is that it's green, which means that we are actually getting water penetration, which means that we can get water depth from this particular data set as well. Additionally, in terms of spatial coverage of ICESat-2 is composed of 6 beams in total. Ultimately, we're getting 3 sets of pairs of beams. The strong and the weak beams, which make up a pair, are differentiated by a 4-to-1 transmit ratio of energy. So ultimately, our stronger beams are getting slightly more data than our weaker beams. Between each pair, we have about 90 meters, and in between each sets of pair, we have about 3.3 kilometers. I'll specify, the way that ICESat works is it flies in one direction, does its little laser, Ashley, if you will, it shoots its laser down and it flies in one direction, which we end up calling ICESat tracks. So we've already talked about the cross-track coverage, but in the along track coverage, we are getting data collected every 70 centimeters or so, which is really great. And in terms of the overall cross-track coverage, we are getting 6.6 kilometers. Temporally, ICESat follows what we call a polar orbit, meaning that it focuses most of its time and energy at the poles of the Earth, which makes sense, right, because of the mission of ICESat-2. It also follows a 91-day repeat cycle, where it ends up creating like a lattice pattern over the Earth, covering the same areas every 91 days. ICESat-2 has been around since 2018 and has a whole boatload of data products that are associated with it. For one, our rawest form of data, our raw telemetry data, is our level 0 data. So this is coming right out of the satellite. The next level down, level 1, is some reformatted data, some converted data, but these are typically used to create some larger products, like our level 2 products, our geolocated photons, and our atmospheric products, which are our normalized relative backscatter profiles. So these are our scientific data sets, if you will. In the next level, we have level 3A and 3B. These are our surface-specific data products. You'll notice that all of these surfaces are related to sea ice, land and vegetation height, inland water, and atmospheric type data products. So while we can get water depth penetration, it was not the intended purpose of ICESat-2. If you were lucky enough to be at the CCOM seminar a couple weeks ago, where Dr. Christopher Parrish, who's online, shout out, let us know that there's actually an ATL-24 on its way, which is specifically created for bathymetry. We're really excited about this, but we obviously don't have that data set right now. So I will be utilizing ATL-03, which is our geolocated photons. So essentially, I'm using X, Y, and Z coordinates for the entirety of this research. So let's look at some ICESat-2 example tracks. This is my favorite part. I'm just gonna let you take this in for a second, because there's a lot of information on this slide. And I'm gonna break it down for you. I remember the first time Kim showed it to me, I was like, oh, what have I gotten myself into? But it's really not that bad. So in terms of this x-axis, we have our UTM northing. So if I took an ICESat track and laid it on its side on a piece of paper, that's essentially what these images are. In our y-axis, we have our depth values. The way that these images on this particular screen are broken down is the top image is the entire track. The following 4 images are broken up quadrants of that track. So if you'll follow my colorization, the first section is this first image, the second image, actually. The second section is the third, and so on and so forth. I'm going to give you a second to just digest this, because I'm going to show you a couple more, and they're not color-coded. I'm gonna take a sip. So let's talk about some really important features that we can see in these particular graphs. For one, we have a clear ocean surface that is showing up in these mostly four remaining pictures. It goes all the way across. It shows up at about 50 meters, which is just our distance from the geoid, which is why we don't expect it to be 0. We can talk more about that if we have questions. We're also seeing things like bathymetry happening in the data, which I would argue starts somewhere around here and continues all the way across the screen. We can also see some bathymetry in this third image as well. It's obviously much shallower, but it's still showing up in our graphs. And then finally, we have some weird things happening in this fourth image. There's some gappage happening. We're not seeing an ocean surface, and we hypothesize that this is because of clouds and fog. We have a satellite traveling all the way down to Earth. We're bound to meet some kind of things in the way, right? So let's look at another example. This is another example of what I would call a really, really good track. We can see a clear ocean surface. We can see some bathymetry happening in here. There's a lot of noise, yes, but this is the nature of ICESat data. How about we look at a bad example? So with some good tracks, we also get things that look a little bit like this. So remember, the first picture is the full track and the next 4 are the quadrants. So we can see a little bit of ocean surface. There's some points scattered underneath. The labels on the side are really weird. We estimate that this is because it was probably collected on a really foggy or a really cloudy day. This is another example of what I would call a really bad track. And this one looks really crazy. We're getting these really dense black blocks happening on the screen. They're happening at places like 500 meters, negative 400 meters. And this is probably because a photon traveled down and got stuck in a cloud and kind of bounced around a little bit. And we obviously got some nonsense data. So it's important, I want to drive my point home here, that with all of our good data, we also are getting some outrageous data as well. So when it comes to processing ICESat-2 data, as you probably can guess, it's a bit of a pain. There's a lot of noise, and because we're collecting data every 70 centimeters, we're getting really large volumes of data as well. Typically, the way that bathymetry is being processed from ICESat-2 data in the literature is through hand extractions. So for example, if I pull up this little piece of track, the way that they're extracting bathymetry is to say, that looks like bathymetry, and then they take it. Where this project differs is that I will be doing all of this algorithmically, completely algorithmically, minimal human power. And I can talk more about how I'm doing this if you are interested, but it's kind of a combination of histograms of depth distributions and some confidence and quality flags from the ICESat data. So why ICESat-2? Before we can talk about the advantages, I'm going to drive home these limitations first. For one, again, because our satellite is up in space and we're traveling all the way down to Earth, we're limited by a couple different things. The deepest possible point that we can get in our data is entirely dependent on the conditions of the ocean. For example, if we look at this picture of Alaska, the water is really turbid and really dark, meaning we're probably going to get a really shallow extinction point happening in Alaska, if any data at all. Whereas in comparison in the Caribbean, we have much less turbid water, much clearer water. We'll probably get our deepest possible extinction point in places like the Caribbean. Additionally, weather is a really big factor in collecting this data. As we saw in some of those example slides, things like clouds and haze can prevent us from getting good readable data. And additionally, we end up getting data that is really noisy and requires a lot of cleaning and a lot of pre-processing. But on the flip side of that, with our advantages or limitations, we obviously have advantages. The fact that we are getting such noisy data is an indication that our instrument is really sensitive, which is really good if we're extracting things like coral reefs and trying to monitor them. So we end up getting data that is really high resolution and high frequency, especially when we think about it in comparison to the way that coral reefs are currently being monitored. It is also free. The data is free and easily accessible. If you download ICESat-2 data online, you get a user's dictionary, you get a data dictionary, you get like an algorithms guide, you get all this support with downloading the data as well. But specifically in the sense of coral reef monitoring, this research and using ICESat-2 has the potential to give us greater spatial and seasonal coverage. Additionally, it will allow us to develop methods that are more reliable, more cost-effective, and also economical. So the research question that this project is going to look at is, can ICESat-2 be used to detect changes in coral reefs? And this is going to be broken up into 3 distinct chapters that kind of build on one another. So our first chapter is going to look at detecting coral reefs using ICESat-2. Our second chapter is going to look at identifying specific hierarchical habitat classifications using ICESat-2. That's a mouthful, we'll talk about it in a minute. And then chapter 3 is going to look at change detection in coral reefs using ICESat-2. So before we can get into these chapters, let's talk about our study site. So we started with the Great Barrier Reef, and my study site of choice is actually Heron Island, which is located in the south of the Great Barrier Reef. This particular study site is composed of both Wistari Reef and Heron Reef. So longitudinally, we get about 16 kilometers in coverage, or in size, I guess I should say. And specifically with these 2 reefs, it is home to 72% of all coral species on the Great Barrier Reef. So for this reason, this makes this the ideal study site for creating a coral reef monitoring methodology. Additionally, the particular structure of both of these reefs is what we call an atoll reef. So inside of these reefs, and I'll point to Wistari Reef, you have some graphics if you want the proper language, but on the inside we have a lagoonal structure which has patches of corals throughout, and on the outside we have a reef edge that kind of slopes down and outwards to the sea. So because of this particular structure, this also makes this the easiest case scenario for identifying coral in remote sensing data. So I do want to note that the results of this research may not be directly transferable to other reef structures, but a possible next step of this research could be to explore how these results do apply to other structures. So this is the particular coverage of ICESat data that I have for this area. I do want to point out, you'll notice there is some overlapping happening here. I don't know if you can see this as well as I can, but for this line, for example, there's like 5 colors here. And I just want to remind you, as we saw in those example tracks, not all data is usable, unfortunately. We are getting overlapping coverage, which is really nice, but it kind of limits what we can do in terms of with the overlapping, if that makes sense. So the first question, the first chapter, is going to look at, can ICESat-2 be used to detect coral reefs? And I plan on answering this by first answering these 2 questions. How do coral reefs appear visually and numerically within ICESat-2 tracks? So here's an example. This is Wistari Reef, and this is a particular ICESat track that goes over. And this is the associated plot that goes with it. So all the way at the bottom of the island, or the reef, we have an orange star which indicates where the reef edge is happening. And if you look in the plot, we have a sloping, really steep collection of data points happening there. We also have a green star in the middle where our lagoon is happening. And this translates as kind of like a choppy surface happening, which happens with the changing of the tides. The depth can change a little bit. And then at the top, we have a yellow star, which indicates another reef edge, which we also get a bit of a sloping line. It's much less steep, but again, we can still see this in the data. We also want to answer the question, which ICESat-2 variables and coral characteristics are the most useful for detecting coral reefs? So we predict that some of these different variables are things like depth, reef profile, slope, and rugosity. Rugosity meaning surface texture. With the exclusion of depth, all of these variables are things that are not explicitly in ICESat-2 data. So these are all parameters that I've had to algorithmically extract from the data. I'm going to tell you about rugosity. If we have this very realistic image of a coral, and we think about how rugosity is measured actually in the water in real life, we use what we call, or not we, but they, use what we call a chain and tape method. So if we imagine we lay a chain on top of these corals, this would be this pink line. We take the ratio of the length of this chain and compare it to the distance that's covered. And so using this ratio, we can get a surface texture value. I obviously don't have a chain that I can lay on top of my ICESat data, so I've created what I'm calling a Windows-based pseudo-rugosity algorithm that is essentially creating a mock of this. So if you imagine I have an ICESat track, I've broken it up into some tiny windows, and I'm connecting the dots between them, essentially. And I'm using this yellow line as my mock chain, and I'm comparing this value to the overall distance covered. And I'm getting some pretty good results. Here's a graphic kind of comparing the different levels of rugosity that I'm getting with what is actually happening underneath the water. So this is a graphic that I've worked on with the help of Dr. Dijkstra, where I'm comparing a category of rugosity values with some photo quadrats of what is actually happening at these particular sites. And they're looking pretty good, which is really nice. So the way that I'm planning on attacking this chapter is as a machine learning classification problem. And I'm going to do this in two different ways, and I'm going to compare the results. So the first way I'm doing this is using a binary logistic regression, and I plan on comparing this to a convolutional neural network. As we know with machine learning, or I hope you know, it is really imperative that we have some kind of ground truth data. So Allen Coral Atlas has provided benthic classifications for these particular reefs, meaning that we can see where the coral is, where the rock is, where the seagrass is, etc. And I've taken this data set and I've created one that looks and mimics the structure of ICESat-2's tracks. So in purple, this is an indication of where there is not coral, and in this like greeny teal color is an indication of where there is coral. So ultimately, the expected results of this chapter are, for starters, an algorithmic methodology for identifying for identifying coral inside of ICESat-2, a statistical analysis on which of these parameters are the most important in predicting the location, and finally, a measurement of the percent of corals within the study area, as predicted by my algorithms. We know in this red box that there is approximately 12% of coral coverage that we can use to compare to the results of my research. I lost that sentence, hold on. Chapter 2. This chapter is going to look at how specific we can identify hierarchical habitat classifications from ICESat-2 data. So this image on the right is an adaptation of the Marine Ecological Classification Standard that I have adapted with the help of Dr. Jen Dykstra to be specific for this particular project. So at the top, we have the coarsest taxonomy, which is our geographic zone, and at the bottom, we have our finest taxonomy, which is our species. From preliminary results of the first chapter, we predict ICESat-2 alone to be able to give us geomorphic structure. But what if we have another data set? Will we get finer taxonomic resolution? We predict the answer to be yes, and we predict that with the addition of some other data set, we should be able to get biological cover, if not percent cover as well. What this additional data set will be determined by the results of the first chapter, but I'm picturing it will probably be a hyperspectral or a multispectral satellite image. So to answer this question, we also need to look at how can we algorithmically differentiate between different biological covers? Meaning, can I tell the difference on an ICESat-2 track between corals, seagrass, sand? I also would like to look at, can we tell the difference between different types of corals? And types is in air quotes because I don't mean the difference between staghorn and flume corals, but I mean the difference between hard corals and soft corals, continuous corals or patchy corals. And statistically, which of these parameters are the most important for predicting these different biological covers? I also plan on attacking this as a classification problem, but I plan on doing it more so either as a random forest or a support vector machine. It may also be a comparison of the two. We're not there yet. But the particular ground truth data set that this is going to use, in addition to the benthic classes that I showed on a couple slides back, they also have geomorphic zones. So I plan on using this in my ground truth data set as well. So the expected results of this chapter are going to be an algorithmic methodology for identifying geomorphic structure and biological cover or percent cover, a statistical analysis on what the most important parameters are in predicting each of these different things, as well as a quantitative solution for how well we can discriminate between different biological covers. And this will also probably look like a total propagated uncertainty model. Chapter 3. Chapter 3 is going to look at detecting changes in coral reefs using ICESat-2. And this is going to build on work by Joan Herman. So Joan Herman looked at assessing the ability to quantify bathymetric change over time using ICESat-2. And in Herman's work, she was particularly interested in episodic change, meaning can we see the difference before a hurricane and after a hurricane? And so her study site is in Florida. This image on the left, the top is her ground truth. The bottom is her results, and the image on the right is the comparison of both of those. And in green, we can see where there's an indication of no difference. So her results look really good. Where my research differs from Herman's is that I'm not interested in episodic change. I'm interested in long-term natural changes happening in the coral reefs. And we know that most of the changes that happen in coral reefs happen because of extreme weather events. For example, in this image, we can see there is a huge difference in the before hurricane and the after hurricane image. I would even say the after hurricane image just like looks sadder as well, but. So I have data of ICESat-2 from 2018 to 2023. So I plan on looking through this data twice a year, once before hurricane season, or wet season, if you will, before November, and once after hurricane season, after April. In order to narrow down these searches, I'm going to identify places along the coral reef that are most likely to have experienced some kind of change. For example, on the outer atoll, we're most likely to see some kind of deposit or degrading of sediment after a hurricane. So I'm specifically going to look there for one example. And I'm going to look for change by statistically analyzing the features that we've previously identified in the 2 chapters prior as being the most important. And I'm going to verify this change by comparing it to photo quadrats that have depth values associated with them. And so I'll tell you, I plan on creating some kind of depth model using these photo quadrats and using the QGIS software to extract similar measurements as the ones that I'm extracting from the ICESat data and comparing them that way. If you want to hear more about that, I'm so happy to talk about it. And the expected results of this chapter, I plan on, for one, producing a robust algorithmic change detection methodology that is specifically designed for coral reefs. In doing so, I'd like to identify the temporal, spatial, and taxonomic limitations associated with this methodology. And finally, in doing so, I would like to create an estimation of the future accuracies and precisions that are associated with the future availability of ICESat-2, because it is still up there and collecting data for us. So in terms of contributions of this research, there are 3 clear deliverables that are being produced by each chapter: a system of detecting corals, a system of detecting geomorphic structure and biological species, and a robust change detection methodology. But the impacts of this research go beyond just the clear deliverables. For starters, we have the potential to create a cost-effective, fast, and accurate system for tracking the health and resilience of these coral reefs. As climate change gets worse, and inevitably it is getting worse, we need systems in place to prevent the further devastation of coral reefs. And this research has the potential to aid in that. Additionally, any new research that happens with ICESat-2, specifically in a new domain, is essential to validating the use of it for other domains. For example, this research will directly benefit the benthic habitat community, the bathymetric and the hydrographic mapping communities as well. And finally, in generating foundational knowledge of the limitations and the advantages of ICESat-2, we hope to inspire more utilization of ICESat-2 across a diverse range of domains, not just ocean-related, not just ice-related. So in terms of my timeline, I am this cute little yellow star happening on May right now, so I'm finishing up the results of my first chapter. I plan on presenting at the end of May at the Canadian Hydrographic Conference those results. I will be starting chapter 2 this summer, finishing that whenever, and starting chapter 3 sometime in the fall as well. And hopefully this will put me on track to defend this time next year. These are some references. If you can read them, gold star for the day. But finally, I just want to say thank you. Thank you for taking the time out of your day for joining me today. Thank you specifically to Kim for letting me come into your office and use your resources and your expertise all of the time, and my committee specifically for your continued support. Thank you to my friends for being here. Thank you to Glenna, who had to listen to this twice last week, and Tyler, my fiance, who has had to listen to this more times than I care to admit. And thank you to my family, who's online somewhere. So with that, I will take questions and comments. Oh, somebody guessed in the comments. It was the Great Barrier. I'm proud of you.
I've heard that people fly lidars around on airplanes. Mm-hmm. Do you think your stuff could apply to that data too?
Potentially, yeah. I think that in terms of the instrumentation difference, there would be a lot less pre-screening probably needing to happen if it's coming from planes, but I do think it would be applicable. Yeah. Do you know somebody?
No, I'm just wondering, like, you were all like, you were ICESat
yes,
all the time, and
yes.
Why not consider your work to be of more general value than
No, you're totally right.
Yourself?
You're totally right. I'm excited about ICESat-2 because it hasn't been used for this purpose yet, so I see kind of an avenue of new research happening with this particular thesis. But you're totally right. Yeah, Haley?
This is a wonderful presentation. I just had a question. You mentioned you want to look at wet season, and I know this is really down the line, but looking at possibly changes in the coral during wet season, would you also look to if there's a period of marine heat wave and potentially take a look at that? Do you think that would even show up in this data?
I do think it would. I think that's definitely something that I should look into. I've tried to narrow down the amount of places and times that I'm looking just because there is so much data and so many places to look. But that is a really good point because you would see probably a ton of coral bleaching happening with a marine heat wave. So really great point. Let me write that down. Yeah. Elias?
Yeah, this is really great. Yeah, well done.
Thank you.
My question it's about like some of the methods like you propose. Like in chapter 1, you proposed CNN and binary logistic regression. My memory serves me correct. Then in chapter 2, you proposed random forest and then SVM.
You have a great memory.
Yeah. So is there any reason for this choice of algorithm? I'm just curious.
There's a science answer and then there's like a personal answer, I guess. My personal answer is that my PhD, I would like to serve as a toolbox of all of my skill set. I maybe plan on working in industry by the time I'm done. And I think that being able to demonstrate a bunch of different algorithms and skill over those is really important. In terms of the first chapter, I chose binary logistic regression because it was the easiest classification algorithm in the sense of I could run it really quickly and see if it would give me results that were worth looking at. A convolutional neural network, a neural network I think is really cool to include in this project, but I think that the convolution of convolutional neural networks could be really beneficial in that we're reading ICESat-2 tracks. I'll tell you more about chapter 2 when I get to it.
All right, yeah.
Yeah.
Like, that's exactly what I thought. I'm like, maybe I'm just trying to, use your practice, try different things, which I think is good.
Yeah.
Well done.
Thank you. Anybody else? Yeah.
I have a question about your validation and training data sets and how you're particularly in those areas where you have a pretty sparse point cloud. How are you dividing your data and using it to then train your model and validate your results?
So I'm using the shape files in order to create I'm using the structure of ICESat-2 tracks and the data files, the shape files that are produced by Alan Coral to get kind of a binary, yes, there is coral here and no, there is not. I'm not training the entire algorithm on the entire study site. I'm just training it on the locations that I have data. Does that answer your question? Oh, and in terms of validation, I have the yes, there is coral and no, there is not. So my classification algorithm, at least specifically for the first chapter, I can speak more so because I've been working there. I'm using it to be able to say, yes, there is coral and no, there is not. Does that make sense? Yeah. Cool. Yeah,
Hi, Gabby. Yeah, thanks for a nice talk. Just building on that question a little bit, I'm curious, I've seen like a particular study site in mind, and I'm curious, obviously the Allen Coral Atlas is very broad as well, so I'm curious where you think you could apply your approach more broadly. Like, it sounds like you're going to focus on this one study site for logistical reasons and other reasons, but I'm just curious, like, based on where the ICESat tracks are, where do you think you're going to have good coverage that someone could then expand on it and use what you have to expand and look elsewhere?
That's a good question. So where do we have coverage? In terms of other reef structures that I think could be really interesting to look at, Hawaii is an example of one because it's a little bit, it's not this barrier reef, but it's more of like a patchy, from my understanding, I'm a mathematician, it's more of a patchy situation happening. So I think looking in Hawaii would be really interesting. We've talked about the Florida Keys as well, looking at those particular study sites. And I know that there is ICESat data in the Florida Keys. I am assuming there is in Hawaii, but it's not something that I've looked into quite yet. And Allen Coral Atlas does a really good job of having coverage of most of the corals of the world, at least the ones that I've looked up so far. They have had coverage. Does that answer your question? Cool, thank you. Yeah?
Are there going to be any theorems in your dissertation?
Ooh.
So far you've talked about developing algorithms. And I'm just wondering if there gonna be like mathy stuff too, or even sort of computer sciencey stuff, or like, yeah, any theorems?
I would argue that there is a lot of computer sciencey, mathy things happening more so behind the scenes. In terms of theorems, I want to say yes. I don't know exactly what that looks like quite now, but I want to say yes. Well, let me get back to you on that. Yeah, good question. Yeah, Val.
You mentioned that the long track sampling is 70 centimeters, and I think I remember right that Chris said that the footprint of each of those beams is like 11 meters, or something much bigger than this room, for example.
Yes.
And I'm curious, you know, the typical size of a single coral is obviously much smaller than that, and they're patchy most of the time. Like you get, even in places where it's dense, you don't get one species that covers everywhere. There's a few exceptions to that. And I guess I'm curious, and I think Chris maybe also said that there were preliminary estimates of uncertainty as a half meter or something like that, which is, you know, pretty big relative to the size of lots of things. And I'm curious whether or not you think that's going to be a challenge to see the signal in that noise that you're looking at, that you're going to have the resolving capability to get at these things?
Yeah, that's a really great question. I think that it's important that I make the distinction between coral reef and corals, because in this particular instance, I'm more so interested in at least the first chapter, detecting where the reef is, and not specifically detecting these individual corals. Because of the limitations that you've mentioned of uncertainty, that's kind of where the second chapter talks about bringing in a second data set, because I am worried about being able to identify one little baby coral happening, which is why I think the research will benefit from something like a hyperspectral image, for example, which I would really like to work with, because there's a lot of information that comes from that particular data set. Does that answer your question? Sort of?
Sorry.
Yeah. Okay.
It's a hard question.
There's a lot there.
Other things. Given the time, I'll even let committee, either in the room or online, ask a question, if anyone has one. Anything in the chat?
Oh, Susie, definitely yes, if you can hear me. She is working for NOAA's Coral Reef Conservation Program. Definitely interested in talking more. That's what the comment says. I can read the whole thing, but I feel like it's not. Essentially, the question is thoughts on weather storms, physical energy, or extreme heat events better targets for your work. I can imagine that satellite images, especially hyperspectral, might see bleaching, but not clear on how these would show up in ICESat data. I'm not particularly looking for bleaching necessarily. I'm more so looking in changes in things like rugosity, depth, slope, those types of parameters. Maybe if coral die and they break with a lag, could be considerable. That's a really good point, which is why I'm checking twice a year. And I do think that that's a good point, that if hurricane season ends in April, that maybe I'm not necessarily looking in April, but I'm looking maybe in June, right before the next hurricane season starts. I do think that's a really good point.
With regard to rugosity, and what your comments made me think of it. In this building, actually in our state of the art, we, in my opinion, totally misuse the word rugosity and how we, and the thing that we miss is that it's scale dependent. And so I've reviewed papers where people report on rugosity, but don't report the scale at which they've measured it. And of course, it means different things to different people. And it may be that the biology discipline has a more rigorous way of talking about that. But I would just offer that you should take care in how you quantify those measurements and what you think they mean at what scale.
Yeah, I think that's a really, really good point. The values that come out of the rugosity algorithm, I don't want to say are completely useless, but they're kind of random, and there's a really weird distribution of those values. So I'm using them more as a categorical classification of what values would represent low rugosity, medium, and high rugosity. They are totally scale dependent though, so that's a really good point and something I'll have to remember when I write this up at some point. Thank you. Yeah.
In chapters 1 and 2, you talked about trying to get at what are the particular features in the data that are responsible for the classifications that are being made by the machine learning methods, if I understood correctly. And I was just wondering if you had thoughts, because you mentioned a couple methods like convolutional neural nets and SVMs that are like don't give up that information very readily.
So I have done separate feature analysis, a feature important analysis. I've also looked at doing, I'm blanking on what it's called right now. It's, principal component analysis. Thank you, Gabby. Principal component analysis, which is obviously very different than feature analysis, but I am comparing the results of those to see if there is any similarities. I've also done covariance matrices to look at correlations between the variables and those types of things.
But would that tell you whether those features are important for the machine learning methods to give the results they're giving?
They, specifically for logistic regression, yes. I haven't gone through the steps of actually doing the SVM yet, so I'm actually not sure. But so far, it has. If you have thoughts and ideas.
I totally agree, but
yeah.
for others, I don't see the connection.
Okay, I'll have to look more into that. Thank you.
We will talk about that.
Yes.
My question's on a slightly different track, which is thinking about where your research fits in the sort of broader landscape of ICESat-2 research and FAIR and CARE principles, if you're familiar with those. The idea that data is open, accessible, findable, reproducible, and you've indicated that the ICESat-2 data is, and I'm kind of wondering what your plans are for making your algorithms open and available to other people.
That's a great question. I would love to. I think that sounds fantastic. I'm not entirely sure what those steps look like. I think you maybe know somebody who does, sort of. Kim and I will figure it out. I would love to publish all of it. I think that is very helpful for the future.
I would be so
yeah, you would talk. Yeah, that would be great.
Join GitHub. That's the start. Yeah.
Yes. Most of it is on GitHub already, which is, I guess, a great start. I am the only one that can see it, though, so that's not. Good question, though.
Anything else? All right. With that, I will thank you for your questions. I have to admit, I had said before this talk with Gabby, I've been to talks where the supervisor jumps in and starts answering questions because they're really interested in all of that. And that's a really, really bad look. But a lot of your questions, I just thought, that's so critical, that's so important, and lots of stuff that we've talked about. So thank you all for being here. I'll ask the committee members online to stay and those who are in the room to stay. And we will have a further discussion. So thank you. Wonderful.